link to your.
Okay, so when we left off last week, we were talking about organelles, and we had left off talking about the endoplasmic reticulum and how there's a smooth version and a rough version. This is part of what we call the endomembrane system, meaning that there is a network of organelles that are all connected together by a series of membranes. Not connected by that membrane directly is the ribosomes. So you have free-floating ribosomes and you have ribosomes that are attached to the ER. The ones that are attached to the ER, when the ribosomes make their product, which is what do they make? What are ribosomes? They make proteins. Yeah, good. So they're going to make proteins. And once they make those proteins, they need to be shipped off they're just really a polypeptide chain at that point. They're not a fully functioning protein yet. So they need to be shipped off to another part of this endomembrane system by way of transport vesicles that are provided for by the rough ER. So the rough ER will put a membrane around, package that protein. They're called secretory proteins. Secrete just means to give off. So they're releasing them. And after they are in that little bubble, that little membrane wrapped around them, they will proceed to the next part of the endomembrane system, which is the Golgi apparatus. The Golgi apparatus is going to take that package, it's going to take that vesicle, and it is going to modify the protein to make it a fully functioning protein. So it says the Golgi apparatus modifies the products of the ER, manufactures some other macromolecules. That would be not something we're going to need to worry about too much. And then when it's done, it has to ship its package too. So once that polypeptide has become a fully functioning protein, now it's got to be shipped somewhere. So the Golgi's job is to receive the package and then do something with it, and then again ship it out after it's done. The Golgi's made up of a bunch of flattened membranes, Sachs lipid membranes, that look like this, kind of look a little bubbly there on the sides. Got a little bubble there. They're going to come from the ER in a little bubble. And they're going to attach to one side of the Golgi apparatus. That side is called the cis side, or the receiving side. That's where the packages will be received. So that bubble will fuse its membrane. It will attach its membrane to the membrane of the Golgi. And in doing so, it then releases all the contents, those polypeptides, into the Golgi. Then when it's in the Golgi, that's where it will be modified. And after it leaves the ribosome, it's really just a polypeptide chain. It's just that primary structure. It doesn't have its secondary structure. It doesn't have its tertiary, or in some cases, its quaternary structure. So in the Golgi, after the Golgi has received it, what it will do is it will make it into a fully shaped, functioning protein. Then when it's done, it's going to ship out of the other side. So it's going to be a one-way direction. It's always going to go one direction. And it's going to form another little bubble that will come out the trans side, or the shipping side of the Golgi apparatus. All right. So super important for making the protein actually functioning it's not going to function right after it leaves the ribosome. It has to be updated or modified in order to do that. And the Golgi is going to be responsible for doing that. Obviously, if there's a problem with shipping, if there's something wrong with the ER and it doesn't ship, that's going to be a problem. It's also going to be a problem if for some reason the Golgi can't receive it. If the Golgi can't pick up that vesicle, if it can't get the package, then that could also cause a problem. Once it leaves the Golgi, it's still in its little bubble. And a very common place that it will end up is staying in the cell as what's called a lysosome. Remember lysosomes? No? When I have freshman biology, I often have the students do an analogy. And on that worksheet that I gave to you, you can do the analogy as well. Lysosomes have digestive enzymes in them. So the proteins that are going to become lysosomes are enzymes whose job it is to break up and digest macromolecules. So it's just it's a sac, it's a storage container of, remember, hydrolytic. If something is hydrolytic, what does that mean? It's going to break it apart with water. It's going to use water to break it apart. So here's an enzyme that's hydrolytic. It's going to use water to break that macromolecule into its constituent monomers. Break it down from its polymer to its monomer structure. Hydrolytic enzymes and the lysosomal membranes are both made in the rough ER. And then they're shipped to the Golgi where they become fully functioning. Okay. They're actually not made by the rough ER so much as they're made by the ribosomes on the rough ER, at least the enzymes are. The membranes themselves will be made on the ER. Some terms, let me shut the door. Keep your intoxicated. She's funny. I hear laughing. I hear her cackling next door. She calls it her cackle too, so I'm not saying anything that I'm not saying anything she doesn't already think. All right. So one of the things that lysosomes do is they will attach to a bubble, a membrane-bound bubble that has trapped food. And that is a food vacuole. So the cell can actually wrap its membrane around a piece of food and bring it in. That process of bringing in a piece of food and wrapping a membrane around it is called phagocytosis. The lysosome, once the food vacuole is inside, the lysosome will attach, or it says fuse here, which is the same thing as attach, to the food vacuole, and then it will release its enzymes, and the enzymes will digest the food that's inside that vacuole. Phagocytosis is a specific form of another type of cytosis called endocytosis. Endo just means to bring in to the cell. So cytocell, we're bringing something into the cell. Phago refers to a solid particle, in many cases a food particle being brought inside. You can also have another type of endocytosis that is called pinocytosis. like Pinot Noir, which is a wine. Pinot refers to liquid. So it's bringing liquid into the cell rather than bringing a solid into the cell. Lysosomes can do the same thing, though. If there's something in that liquid that need to be broken apart, they will break it apart. Harper?
Like, does every endocytosis result in it, like, breaking it down, or is it just...
That's a great question. So Harper's question was, does every event of phagocytosis mean that whatever's brought in is digested? And we don't think so. And that actually kind of leads me into my next question. So hold that thought for just a second, because I'm going to come back to it, I think, in the next slide. The last thing I wanted to say was lysosomes also will break apart their own macromolecules and their own organelles. If an organelle is shot, it's just done, it's old, it's worn out, it needs to be recycled, that's the lysosome's job. Lysosome will break it down into monomers and polymers, same with macromolecules, so that those pieces can be reused. Because just because mitochondria doesn't work anymore doesn't mean we don't still want the lipids and the proteins and all the parts that are in there. We still want to use those things. And then these are pictures of that whole process of bringing in a food bubble. So they're wrapping it around. This is, let me do my food in a different color. Here's my food. My food's going to come inside, and my membrane is going to completely wrap around it here. My food's in there. And then this lysosome comes along in purple, and it will digest it. And there's a lysosome fusing with a mitochondria. Okay, it's not the next slide, but we will get to that point. Sometimes things stay inside of the cell after they've been brought in. And we'll see that when I'm going to talk a little bit more about chloroplasts and mitochondria and why mitochondria. We think that mitochondria and chloroplasts actually at one point were living on their own. And a bigger cell wrapped a membrane around them and brought them inside. And rather than digesting them, they, for whatever reason, stayed there. And that's how we got today's eukaryotic cells. And there's some evidence for that. And I'll talk about that when we get to it. But I got a little ahead of myself, I think, talking about it. It's to come. Another organelle that we need to talk about is the vacuole. Vacuoles are, they come from both the ER and the Golgi. They're just really large vesicles, so really large membranes. And they can do all sorts of different things, depending on what species you're talking about and depending on what cell you're talking about, even within the same species. There can be, like I said, there could be those food vacuoles that are brought in by phagocytosis. There are also central vacuoles that you find primarily in plants, and that's going to be just a really large, kind of like tank for water, usually. It could also be for things like for sucrose, for holding sugar as well in plants. We also have vacuoles in our cells, but they're pretty small comparatively to plants. Plants usually have a large central vacuole, and if you see one really large within a plant cell, you see one large structure that is not the nucleus is probably going to be the central vacuole. The other one's kind of fun is called a contractile vacuole. And these guys, their job is, and we'll talk more about this when we get to later in the unit, we get to osmosis and diffusion. But There are some little critters that are single-celled. This is my little cilia around my little single cell. And what happens is over time, they're a little leaky, meaning that they live in water and they take in water by accident. They don't really want that water, but it comes in. It's like there's openings, and we'll talk about exactly why that is a little bit later. But point is that if they continue to take on water, what would happen to them? Probably they would explode. They would pop. Okay, so they don't want that to happen. So when they get enough water into them, it's stored in a little vacuole. And when that vacuole gets large enough, it squeezes the water back out. It contracts the vacuole. And as it does that, the little paramecium, in this case, this little microorganism, it like scoots along. So it uses it as a way to propel itself through the water. It squeezes the water out, and the water just goes one way, and the little critter goes the other way. It's kind of a cool little feature. All right, let's see, what is that? There's the central vacuole. In a plant cell, you can see it's really large. It's even larger than the nucleus. The nucleus is over here. Sometimes it'll take up more than half of the cell. Plants can't drink, so it's important for them to store water whenever they get it. Whenever it rains, they're going to take in that water if they can. Okay, that's just great. Here's kind of an overview of those whole relationship, the endomembrane system. I've got that's not a good color. Let's use yellow. I've got my little ribosomes here. And the ER is going to put a membrane bubble around those proteins. That was a bad color. As they get shipped to the Golgi, they go in the Golgi on the cis side, fuse their membranes with it. They continue through to the trans side. As they do so, they will become fully functional. They get their secondary, tertiary, and quaternary structure, and then they get shipped back out. They can either stay inside, and that's this little guy here. The purple one is supposed to be a lysosome, or they can get shipped out. Some proteins get shipped out. They'll float around in the bloodstream or the rest of the body. Questions on that? All right. Let's talk about two other organelles that are not part of that endomembrane system, called the mitochondria and the chloroplasts. And these guys are both involved in energy in one way or the other. Mitochondria is going to be the site of what we call cell respiration. We'll have an entire unit on cell respiration. It's basically the process of taking your food and oxygen and turning it into energy. And our energy source that we're going to talk all about in detail is ATP. Okay, whenever we talk about energy, our body needs ATP. Our body does not use our peanut butter and jelly sandwich directly. Instead, what has to happen is it has to get converted into cell energy, and that cell energy for us is ATP. And for every living thing, it's ATP. Mitochondria converts our food into ATP. Chloroplasts. We do not have chloroplasts. Chloroplasts are found in plants only, as well as algae. And that's where photosynthesis is going to take place. Photosynthesis is going to be where we take sunlight and carbon dioxide and convert it into food for the plants, and by extension, also food for us. Then the last one that's also listed on here and for some reason is not bolded. Let's bold that. Is the peroxisome. And we'll talk how those are related as well. Okay, so we will spend a lot of time on both photosynthesis and cell respiration. They're actually in the same unit. I think that's 3, unit 3, maybe. Wait, is it that soon? Maybe it's 4. It might be 4. Anyway, it's coming. Coming to an AP Bio class near you. But back to what Harper's question was, when something comes into the cell through phagocytosis, does it get immediately digested? There are 2 cases that we pretty sure that this phagocytosis happened, and what got brought in did not get digested, and that's mitochondria and chloroplasts. And we think that because mitochondria and chloroplasts have a lot of similarities to bacteria, or remember, that's our prokaryotic cells. Those are the ones that have the ribosomes, they have the cytosol, they have a cell membrane, but that and DNA and a nucleoid, but they don't have any organelles. Mitochondria and chloroplasts both have a double membrane, not one membrane, but 2 membranes. Why might they have 2 membranes? Why might that be? I'm a little mitochondria. And I have to, if I'm living on my own, I have a membrane. And then if something else comes along, let's make it Pac-Man. Anybody like Pac-Man? No? Okay. And then he eats him. How many membranes am I going to have now? I'm going to have 2. If I come inside here. Oh, now I'm going to have a second membrane. Okay, I'm going to have a second membrane. So they have double membranes. They also have their own ribosomes. Weird. Why would mitochondria have their own ribosomes? Unless they were on their own, making their own proteins. They have circular DNA. Eukaryotes do not have circular DNA. You know what kind of DNA we have? We have linear. Right, ours is like this. But that's not what you find in a mitochondria. You find circular pieces of DNA. They grow and pretty much independently reproduce on their own into cells. So if they need to, they can reproduce without the cell reproducing. So we'll talk about mitosis and how a cell makes a copy of itself. Mitochondria can actually do that on their own. They don't have to wait for the cell to reproduce. So that's all led to what we call endosymbiont theory, which is the idea that, think about it, wouldn't you love to have a chloroplast inside of you? I guess. What would you be able to do then? Photosynthesize. You'd be able to make your own food. So the theory suggests that what happened was there was an early ancestor of eukaryotes, and it ate either a mitochondria or a chloroplast. And there became a relationship. Rather than getting digested, it's like, hey, I'm keeping this thing around. It's kind of useful. I mean, there was no like cognitive actual thought like I'm making it sound, but mitochondria made energy for the cell, and chloroplasts made food for the cell. Why not keep it around? And those pieces of evidence are the reasons why we think that. The fact that they have their own DNA, the fact that they reproduce on their own, the fact that they have double membranes, all is strong evidence for the idea that they came from They were on their own. And then they got gobbled up. There's a little mitochondria getting gobbled up. There's a little chloroplast getting gobbled up. All right. Okay. Bell ring at 17? Say. What is it? 19. All right, let's look. See what else we have then. Oh, yeah. No, I don't want to go into the next one because we've only got a couple minutes. All right, let's stop there.